Chapter 9, slide really 27. Now, most of this information is found in slide 60 through 63. So this is a summary, slide 60 through 63. So with your neuromuscular junction, you have a synapse. When your nerve impulse arrives at the axon terminal, acetylcholine is released and binds to receptors on the sarcolemma. This causes sodium and potassium to flow, but more sodium goes in, so the inside of the cell becomes less negative. This is a graded potential of local depolarization. If that is strong enough, then you can reach threshold to open up voltage-gated sodium channels. Sodium rushes into the cell through fast voltage-gated channels, and it will depolarize the cell. Those channels close and slow voltage-gated potassium channels open. And potassium flows out, doing repolarization. So it restores the resting membrane potential. It restores the resting membrane potential by changing the electrical gradient. Chemical gradient has to be restored by the sodium-potassium pump. The action potential goes across the sarcolemma, down the T-tubules to the SR. Calcium's released. Calcium binds to troponin, which moves tropomyosin, lets myosin bind to actin, and use ATP to move the actin towards the middle. You do not need to know the details of the cross-bridge cycle. Don't worry about detachment, cocking, power stroke, recovery stroke, or any of that. Here we are at the summary. Be able to define motor unit. Slide 64. Understand a twitch, which is a single stimulus. But more importantly, understand tetanus, incomplete or fused tetanus, or incomplete, unfused tetanus, or complete fused tetanus. To do a bigger job, recruit more and bigger muscle fibers. Be able to define muscle tone, that it's due to spinal knee reflexes. You need to be able to discuss the 4 ways that you have power for the muscle. You have stored ATP, the creatine phosphate. You can perform anaerobic glycolysis and aerobic respiration. So creatine phosphate, anaerobic pathway. You need to know the lactic acid that's formed, why it's formed, or what you have to do to get rid of it. Essentially oxygen debt. Aerobic pathway, you have to have oxygen and you get lots more ATP from it. Understand oxygen debt and how you need to get out of oxygen debt.
It seemed like the whole test right here. Took my name so long.
Understand the concept of the resting length relationship between tension development. I'm probably not going to test you on that, but we don't have as much material on this test as we usually would, so I may ask more in-depth questions. I don't have to do that.
No.
You need to know the 3 muscle fiber types. And the basics of how they work. Know what endurance exercise does for you and what resistance exercise does for you. And remember, when Anneline's muscles get bigger, it's because each muscle fiber is getting bigger.
Right.
For smooth muscle, you need to know that you get external sources of calcium from these caveoli. And you need to be able to define peristalsis. Now, there are a lot of gap junctions between smooth muscle cells. What are gap junctions, you might ask me? Gap junctions are channels between the cell that allow fluids and ions to flow through, as opposed to the desmosomes, which are structural rivets, and the tight junctions that make them waterproof. Gap junctions allow electrical and electrochemical signals to pass from cell to cell, enabling these smooth muscles to work as a single unit. So here we have gap junctions that allow the ions to go through from cell to cell. You can have pacemaker tissue in smooth muscle. And although calcium is still the final trigger, it acts through binding to calmodulin and a different pathway. The initial reaction to stretch or stress is to contract. The nervous system. You need to know the functional and structural divisions of the nervous system. And if you'll recall, we take the nervous system, divide it in two, one of the two gets divided in two, one of the two gets divided in two, and one of the two gets divided in two. Basically giving you this diagram for the nervous system. 2 subsections. One of them's divided into 2 subsections, one's divided into 2, one's divided into 2. You need to know the various levels of the nervous system divisions. You can expect to see a large number of questions about the functions of the various neuroglia. astrocytes, microglia, ependymal cells, oligodendrocytes in the central nervous system, and then Schwann cells and satellite cells in the peripheral nervous system. You need to know the main function of each one of those cells.
So can you repeat that last one?
You need to know the main function of each one of those cells. Each one of the neuroglial cells. The neurons, you need to be able to label the neuron. You need to know that the neurons have to have a bunch of oxygen and glucose in order to function.
What in glucose?
Oxygen.
Okay.
They have a high metabolic rate. You need to be able to define dendrite and axon based upon the conduction. A dendrite conveys electrical signals towards the cell body, and they are graded potentials. And if you'll recall, the graded potential is an electrical charge that is just moving through fluid that will get smaller as it goes further away. It's not reinforced by voltage-gated channels. So it's a local voltage change that will dissipate that's not reinforced by voltage-gated channels. The axon will produce action potentials, which are always the same size. They're discrete units. They are reinforced by voltage-gated channels. You need to know the function of the myelin sheath. You need to know the function of the nodes of Ranvier. Be able to define white matter and gray matter, and then be able to define clusters of gray matter in the central nervous system or the peripheral nervous system, or basically give their name, nucleus or ganglion. The same classifications of the nervous system can be applied to the functional classification of neurons, sensory, motor, and the association or interneurons. You still need to remember all of the voltage versus the chemically gated channels. And depolarization and repolarization, but also hyperpolarization. If you hyperpolarize the membrane, the cell is going to be less likely to generate an action potential. If you depolarize the membrane, you're going to be more likely to produce an action potential. So excitatory versus inhibitory. And then, of course, your action potential, again, but there is a hyperpolarization phase. That means that there is a brief period of time afterwards in what is called the relative refractory period, where it's less likely to happen. There's also an absolute refractory period, means it's impossible to generate another action potential. Know the effects on axon diameter and myelination for conduction velocity. And you need to have a basic understanding that the myelin sheath speeds up the nerve conduction velocity. Now, we talked about the synapse. We've got a presynaptic neuron and a postsynaptic neuron. And we talked, I wrote up on the board, EPSP and IPSP, right? Excitatory postsynaptic potential and inhibitory postsynaptic potential. So we saw the video that outlined the information that is on the following slides. Your action potential arrives to the axon terminal. Calcium goes in and causes exocytosis of the neurotransmitter. Binds to channels, it'll cause ions to flow, either inhibiting or exciting. Has a graded potential. Here you see the 3 different ways that you can get rid of the neurotransmitter and stop its activity in the cleft. And here we see EPSP and IPSP. So you should be able to tell me if I asked, what will happen if potassium flows out of the cell? Will it be more likely, less likely, or the same likely to produce an action potential?
Less likely.
Potassium flows out of the cell?
Yeah,
Less likely.
less likely. So you need to understand the concept of your cell membrane, what ions traveling which direction, what will happen.
Sodium travels in, right?
What?
Sodium travels in? The sodium travels in?
Sodium coming in will cause what?
Depolarization? Wait.
Depolarization, which means it's what, more or less likely to generate an
More
action?
likely. Wait.
Yes. Yes, more likely.
More likely.
Okay. Alright.
The whole point is, are you getting closer to threshold or further away from the threshold?
Right.
Wait. I did.
What?
Wait. Does potassium bring you back up to the threshold after you have
No. Potassium brings you back down. Potassium's going out. The inside becomes more negative, so you're going down.
the sodium
Away
would
from
bring
the
you
threshold.
to action, meaning
Yeah.
threshold, and then that would bring you.
So we're talking about graded potentials. We're not talking about the depolarizing and repolarizing of an action potential. We're talking about local graded potentials that are depolarizing in nature or hyperpolarizing in nature. Depolarizing in nature means they're more likely to generate an action potential eventually if you have enough of them. So it is an excitatory. Inhibitory is going to be less likely because you're going to hyperpolarize, get further away from threshold at that moment. So this is a no vote. Remember we talked about voting. Some signals are yes, some signals are no. Inhibitory is a no vote. You take a vote in the class and you make your decision. Did we get to summation?